Hey everybody, today we're going to be talking about meiosis and meiotic mistakes. You'll recall that somatic cells are your normal body cells. They have two copies of every chromosome, one from the biological mother and one from the biological father. Because they have two copies, they are diploid. These pairs of chromosomes are called homologous chromosomes. Homologous chromosomes are similar in size, structure, and coded information, but they are not identical. Gametes are your sex cells. Those are the cells whose job it is to make a new organism. After fertilization, each chromosome will eventually become one of the homologous chromosomes that make up the diploid offspring. Mitosis is what somatic cells do to make more of themselves. This process duplicates the DNA exactly and results in two identical daughter cells. All of the cells involved are somatic, which means they are 2N. They have two full sets of chromosomes. So how do we make sex cells? Sex cells start as a diploid cell, and the end is four non-identical daughter cells, which are all haploid. To do this, the cell has to go through two rounds of division. The first round is called reductional division. This is where the cell goes from diploid to haploid. It is called reductional division because we're reducing the number of copies of chromosomes in each cell. We also call this round Meiosis I. Meiosis I is where all the new stuff happens that's different from mitosis. We reduce the number of chromosomes and we also make the daughter cells genetically different. Meiosis II is the second round of division in meiosis. It is called equational division and results in the four non-identical daughter cells. There are two important roles for meiosis. The first is to produce the gametes for sexual reproduction. The gametes must be haploid, so when they meet up and fertilize another gamete, the resulting offspring is diploid. The second role for meiosis is to create diversity in the species. This diversity helps the species survive by making sure that each individual is a little bit different. There are three mechanisms for creating diversity: crossing over, independent assortment, and random fertilization. Here's a meiosis overview. The diploid cell proceeds through the cell cycle as normal. This figure shows a cell with one pair of homologous chromosomes, so 2N equals 2. The DNA gets replicated in S phase, and the DNA is shown here as sister chromatids. Remember, the sister chromatids are genetically identical, so they are shown in the same color. The homologous chromosomes are not genetically identical and are shown as either blue or red. At the end of G2, the cell enters meiosis. During meiosis I, instead of the sister chromatids being pulled apart like we saw in mitosis, the homologous chromosomes are pulled apart. Now, each resulting cell gets either the blue parent chromosome or the red parent chromosome, but not both. The daughter cells are haploid. They are haploid because they only have one copy of each chromosome, either the red or the blue. In meiosis II, the daughter cells divide again, and the sister chromatids are finally separated, each daughter cell making two of their own daughter cells with single chromatid. Each step of meiosis has four phases. They should all look familiar to you. We are missing one phase from mitosis, and that is prometaphase. Mostly the same things are happening in each phase as we saw in mitosis, with a couple of exceptions. Starting in meiosis I, the homologous chromosomes are going to line up very, very closely in prophase I. This is called synapsis. They line up so closely that sometimes they wind around each other and attach to each other. The place where they attach to each other is called the chiasma. The chiasma allows some genetic material from the red parent's chromosome to swap places with the blue parent's homologous chromosome. So you might end up with a homologous chromosome that was originally from the blue parent carrying a little piece, or maybe a lot of pieces, of genetic material that came from the red parent. This all happens during prophase I of meiosis. If you follow this to the logical conclusion, each daughter cell will be genetically different because none of them will get exactly the same genetic information from the red parent or the blue parent. In middle prophase I, The centrosomes are going to move towards the poles, and the spindle fibers will form, and the nuclear envelope starts to break down. In late prophase 1, we have the microtubules are attaching to both kinetochores on the centromere. This is different from what we saw in mitosis, where both poles attached to the same chromosome and pulled the chromatids apart. In metaphase 1, we see the second mechanism that increases the genetic variability in eukaryotes, independent assortment. Remember, during meiosis 1, the homologous chromosomes are pulled apart. Which way they line up on the plate is random. This cell has n equals 2. It has two different chromosomes, one set of each from blue parent and one set of each from the red parent. On the left, the blue parent's chromosomes are both on one side of the metaphase plate. During anaphase 1, when the homologous chromosomes are pulled apart, this will result in all of the blue DNA going to the left and all of the red going to the right. It is equally possible that the blue parent's chromosomes don't line up together. The cell on the right shows another possibility for arrangement. If the chromosomes line up in a mixed fashion during metaphase 1, then during anaphase 1 they will be pulled apart, and the resulting daughter cells will have some genetic material from each of the parents. In this way, independent assortment introduces a huge amount of genetic variability into our gene pool. There's a formula that predicts the number of different gametes that can be produced by independent assortment alone, 2 to the n power. In the case of humans, n equals 23. 2 to the 23rd power is 8.3 million different possibilities. Let's do a couple of problems. If your chromosome number is 2, the formula 2 to the n gives the number of different gametes. So for an n of 2, you can have 4 different possibilities. If your chromosome number is 4, however, 2 to the 4 would give you 16 different possibilities just from independent assortment. In anaphase 1, the homologous chromosomes will move apart towards the opposite poles of the cell. Remember, each of the microtubules will attach to both sides of the sister chromatids and pull the entire homologous chromosome towards one side of the cell. Telophase is when the chromosomes have finally reached the poles and the spindle fibers start to break down. You're going to get reformation of the nuclear envelope, and cytokinesis will happen as normal. In meiosis II, it's essentially the same thing as mitosis. There is no duplication of DNA that happens here. We are going straight into meiosis II from meiosis I. We're going to be splitting up those two daughter cells. There are 4 phases to meiosis II. The difference here is that in anaphase 2, we're going to pull apart the sister chromatids, so each daughter cell will end up with a single chromatid. The third mechanism for genetic variability happens during fertilization, and this is basically which gametes are going to meet each other. If you have 100 million sperm cell trying to fertilize an egg, you have a very small probability of similar sperm fertilizing different eggs. This process, like all biological processes, sometimes doesn't happen perfectly. One of the most common mistakes is non-disjunction of the chromosomes. This is when the chromosomes don't separate properly. This can happen in anaphase 1, when the homologous chromosomes are being separated, or during anaphase 2, when the sister chromatids are being separated. If a non-disjunction event happens in meiosis I, all 4 gametes will have the wrong number of chromosomes. 50% of them will have an extra chromosome, and 50% are going to be missing one chromosome. If the non-disjunction event happens in meiosis II, half of the gametes will have the wrong number of chromosomes. 25% have one extra, 25% are missing one, but 50% of the gametes will have the correct number of chromosomes. When one of the gametes resulting from a non-disjunction event is used during fertilization, it is called aneuploidy. This results in a zygote having an unusual number of chromosomes. When the zygote is missing an entire chromosome, it's called monosomy. When the zygote has an extra chromosome, it's called trisomy. Trisomy 21 is one of the more common non-disjunction events that results in a viable fetus. This condition is called Down syndrome. You can see the extra copy of chromosome 21 on this karyotype. Trisomy 16 and 18 are also not uncommon. In fact, trisomy 16 is the leading cause of miscarriage due to atypical autosome numbers. Trisomy 18 is the cause of Edwards syndrome and is invariably fatal. Only 1% of children with this syndrome make it to 10 years of age. That's it for today. See you in class.